Alright, how y'all doing? Just as a video to show you a better explanation of prokaryotic cell parts and cell organelles. Some of these you've had before, I'm sure, and some you haven't. So I'm just going to go over a couple of these in more layman's terms, assuming that you know nothing about these. So the first thing we see here that this bacterium, which is what we call one bacteria, this is shaped kind of like a hot dog. And so the shape of this particular kind would be called bacilli, and there's other shapes as well. So don't think that all bacteria have this shape. So let's start kind of like from the outside in. And the first thing that we see, the longest, and this is probably a word that you do know, is flagella. And these are basically used for locomotion. It's how the bacteria um, travel around. Some of them have them, some of them don't. Typically bacteria that live in very aqueous solutions tend to have flagella, but not all do. And they also have different arrangements. This is called Lophotrichus, but there are some bacteria that have them all over, some have them on both ends, some only have one. But flagella are used for movement, and so on your sheet where you're studying, that's a good word for there. We have this other word now called fimbriae, and these look like little fingers, and that's kind of a good analogy for them because the bacteria use fimbriae for kind of feeling their way around and sticking to things. And so, if a bacteria lands on a table, one of the ways they're going to be aware of the fact that this probably is not a good place to reproduce is that in part the fimbriae is going to, in essence, give them signals or lack of signals that there really is no food outside. And so that's part of the way. They also use something called a glycocalyx as well. But generally speaking, the fimbriae used to hold on to stuff and to kind of sense the environment. What's here, what's not. As certain salts and foods diffuse in and out, the bacteria then can decide whether this is an area that it should begin reproduction. It's also why, for example, restaurants and public doors are made of stainless steel, restaurant tables, partially because the bacteria, it's very difficult for them to stick onto such a non-porous surface. All right, so enough about that word. So if we get in a little more, now we have these layers, and we can see here that there's kind of several of them. And bacteria don't necessarily have all of these, so I'm just going to cover some of them. A capsule is generally outside of the cell wall, and it can be of various thicknesses. It can be used to kind of protect the bacteria from the environment. Next one in is called the cell wall, and that's pretty important. Almost all bacteria have a cell wall, some don't. So basically, the idea with this is no different than a plant cell. It's a rigid kind of layer of protein, per se, that kind of gives it shape and structure, and it's very rigid. And so really what it does as well is it protects this thing from osmotic pressure, which means that it allows the bacteria to not break or crack in certain situations. And it's also, by the way, the major site of where most antibiotics work on. And that's why when you take an antibiotic, it's one of the ways the antibiotic, if you've ever wondered how they know how to kill the germs and not your cells, human cells don't have a cell wall. And so that's in part why some antibiotics work. Inside of that one is called the plasma membrane. I'm sure you've had that before. That's a semi-permeable membrane. That's the one that allows certain things to cross in and out, and it has proteins embedded in it. And that is one of the ways that the cell can regulate materials going in and out. It kind of separates the outside from the inside. And I would use an analogy that's kind of like the human skin. So if we move in more, we have... and I'm not going to cover all these. Some of these words I'm not going to cover, you're not going to see necessarily. So we do have other areas, cytoplasm, also cytosol. There are two words, but they're generally used the same. That's kind of like the liquidy part of the cell. It's where the reactions happen and whatnot. That's not particularly difficult. But these little dots are very important. And you can see these little dots all over here called ribosomes. And these are where the bacteria will produce its proteins. And of course, how does it know what proteins to make? It needs to use this stuff in here called the DNA. And chromosome, basically most bacteria have one chromosome and it's a circle. And you'll note that there is no nuclear envelope, there's no major membrane-bound organelles. So the ribosomes basically make the protein, and the DNA, of course, codes for it. That makes a lot of sense. Some bacteria have this little thing here. And this is a little extra piece of DNA called a plasmid, and they don't need it, and so they're not required for living. but this is a lot of times where bacteria can shuttle different genes back and forth across to each other. And that's one of the reasons why, most likely incorrectly, you've learned in high school or before that bacteria are all clones and they all reproduce by binary fission. They're all the same. It's so not the case. So this is where bacteria can have some extra genes, and they can literally give them to each other. Nuclear you don't really need to cover. So I think that's a pretty good summation of the parts, at least to give you a start of what they do. And of course you'll need to know these in more detail. Do not worry about inclusion at this point, but hopefully that was a good start to help you out with the general structure of prokaryotic cells.